Immunodiagnosis of infectious disease. Serology is the study of serum. If you recall, serum is the liquid portion of the blood that is free of clotting proteins. So we take a sample of blood from a patient, put it into a tube. That tube is allowed to clot, and then we put it in a centrifuge, and the centrifuge spins really, really fast, pulling all of the solid substances to the bottom of the tube. Those solid substances include all of the cells in the blood, as well as all of those clotting proteins. And then what's left on top is serum. And serum contains all of the other proteins that are found in the blood, including antibodies. So in serology, it's the study of antibodies and their reaction with antigens in the diagnosis of infectious disease. Antibodies are the proteins in the serum that bind to antigens. So this is what a typical antibody looks like. And where do antibodies come from? When your body gets exposed to an infectious disease, or something that shouldn't be in the body, sometimes that starts a local type of inflammation and infection. And if that local type of inflammation stimulates the white blood cells called B cells to proliferate, B cells will then be activated against a specific antigen. Once B cells are activated, they're going to divide like crazy and start turning into plasma cells, which pump out large concentrations of antibodies. So initially, when you get exposed, you have a small amount of antibodies, and it takes a really long time for those antibodies to be developed and to reach high enough levels. So if we're looking for the presence of antibodies in someone's serum, it may take a while after they've been exposed to the infection or the microorganism to actually develop those antibodies. But after a first exposure, the second time they get exposed, they're going to develop a large number of antibodies very quickly, because those cells will be prepped and primed and ready to go to mount an attack. And that's kind of the basis for vaccination and all that other type of stuff. So antibody, that's what our B cells produce, and those are floating around in our serum right now. Antigens are anything that causes an immune response, usually high molecular weight proteins or polysaccharides. So an antigen is this smaller substance. The antigen would bind to the antibody. The antibody binding site is extremely specific for certain types of antigens. So you can see all these different types of antigens, and the only one that fits in the binding site is this one right here. There are two basic types of immunologic tests. The first one is when we use known antibodies. So we have an antibody that specifically recognizes COVID, or an antibody that recognizes human chorionic gonadotropin, HCG, in a pregnancy test. Sometimes we fix those antibodies to a synthetic medium. So we have a little piece of paper on the bottom, filter paper, nitrocellulose membrane, and then we have the antibody that's been stuck to it. And that antibody has its little antibody arms sticking up in the air. So it's stuck to a piece of paper or some type of membrane. Then we get a patient sample, and we want to see if the antigen is actually in the patient sample. So we will put that patient sample over the antibodies, and if that antigen is present in the sample, it will stick to these antibodies. So going back to this picture here, we're going to put patient sample around the specific antibody. And if the patient sample has what we're looking for, has the antigen that we're looking for, then it will bind to the binding site on the antibody. And then that's detected by a color change or by a series of events that we will talk about as we go through here. An example of that type of test is a COVID test. So we have antibodies against COVID. If the patient has COVID, they're going to have COVID antigens in them, and the antibodies that we know are against COVID will combine with those. The other type of test wants to see if a person has been exposed to an infectious agent, and it will look for the presence of antibodies in the patient's serum. So we use known antigens and we put those antigens on a piece of paper. So we put the chunks of the COVID or the chunks of the HIV on the paper. Then we're going to take the patient's serum and we're going to flood that paper with the patient's serum. If the patient has antibodies against that infectious agent, the patient's antibodies are going to bind to the paper. And through a series of washing steps, they're going to remain bound because that's a pretty tight binding reaction. And then we have detectors that will be able to pick up on whether they had the antibody or not. So in one aspect, we're testing the patient's specimen for the presence of the actual pathogen or the actual antigens. In the other aspect, we're testing the patient's serum for the presence of antibodies against that specific antigen. So here you can see a breakdown of the basic types of immunologic tests. So the antigen is on the paper. We have a primary antibody against it, and it's linked to a substrate. So if we put the patient specimen down here on the paper, if the patient has that antigen in them, then we would use this specific labeled antibody to detect it. And if we see a color change, we know that the patient had that antigen in their serum or in their sample, wherever we took the sample from. Here in indirect ELISA, we have an antigen down at the bottom. Then we use a primary antibody, a secondary antibody, and that secondary antibody is linked to a substrate. In this instance, we have a capture antibody. So this is a known antibody that will then capture any antigen that's found in the patient sample. And then it'll bind to the primary antibody, secondary antibody, and create a substrate. We don't really use the competitive ELISA too much in clinical practice. So we're going to focus on these guys here, and we will be performing an ELISA once we get to our basic immunology course. So you can look for actually being able to perform these in that course. Immunodiagnosis of infectious diseases. So they're powerful tools and excellent for detecting infectious agents. However, like I mentioned previously, if this is the patient's first time getting exposed to something and you want to see if they have been exposed, it usually takes 10 to 14 days to produce an immune response, and sometimes longer. Immunocompromised patients also may have a diminished or non-existent response, so you may not see antibodies at all even though they've been exposed. Cross-reactive antibodies can also yield false positive. Cross-reactive means that you have one antibody that's supposed to recognize one specific thing, but it happens to recognize and bind to multiple things. So an antigen, again, those are the antibody binding properties of the molecule. So those are the proteins or polysaccharides or different substances that antibodies are actually going to bind to. An immunogen is the portion of that molecule that actually elicits an immune response. It's going to elicit the production of antibodies by B cells. Epitope is available antigenic determinants. There's polyclonal and monoclonal antibodies. Polyclonal is a mixture of multiple antibody molecules derived from multiple cells against multiple epitopes, and they're found in an antigen or directed against different antigens altogether. Immune complex, the antibody attaches to the antigen, often written as Ag-AB immune complex. Acute and convalescent antibody titers. Acute phase antibody titer happens within 1 week of manifestation of symptoms. Specific IgG is usually not detected in serum collected in this phase. Convalescent phase antibody titer, the recovery phase, usually 2 weeks after the acute phase. Significant increase in IgG detected in this phase is for diagnostic of the infection. So IgM switches to IgG. Primary and secondary responses. Primary response is immunoglobulin M or IgM. The detection of IgM is noted as primary current or recent infection. So if we investigate patient's serum and we see that there's IgM antibodies, we assume that it's the first time they've been exposed. They are currently infected or they had a recent infection. The secondary response is usually the subsequent exposure, and that's immunoglobulin G, IgG. It's known as an anamnestic response. The presence of IgG antibody alone suggests previous infection or exposure. So during the primary exposure, this is the first time you've ever been exposed to the antigen. You're going to see an increase in IgG. The second exposure, you see a lot of IgG produced. When we look at IgM, the first time they're exposed, it increases earlier. So you can see a peak earlier than IgG. And then the second time they're exposed, there's a lesser peak. Patient serum is tested for antibodies to specific antigens taken from an infectious agent. Each patient's serum is unique, which is why we do a serial dilation to produce a semi-quantitative value of concentration. A standard amount of antigen from the infectious agent is mixed with each dilution, and we report it as an antibody titer. Some infections simply require only IgG to be present, like rabies or HIV. Others may require interpretation as to acute, subclinical, or past infection. So some infections, it's just they had it or they didn't. Other infections, we would like to know a little bit more information. Is it happening right now? Did it happen in the past? And how can that guide our treatment? Monoclonal antibodies are antibodies that are derived from one cell that has been exposed to one epitope. The cell divides, producing a clone of identical cells. It produces antibodies specific to one epitope, high specificity and strong avidity. Antibody specificity and cross-reactivity. Antibodies are usually very specific, but antibodies can bind to similar epitopes from other organisms. That causes cross-reactivity, and those are known as heterophile antibodies. False negative and false positive. A false negative means that it was a negative result, but it should have been positive. So it's falsely negative. So negative result for a patient who's actually infected. Mutated antigens, immunocompromising conditions may result from the competition of IgM and IgG antibodies, or we may have low sensitivity of the test system. So if we don't have enough of the patient's specimen, then we may not be able to detect what's in it. False positive is falsely positive. So it's a positive result for a patient that was not infected. This may result from the presence of rheumatoid factor, cross-reacting antibodies, or the reactivation of latent organism. Immune status testing. This is testing how many antibodies you have against a particular thing that you've been vaccinated against. Hepatitis is a big one that they test for your immune status. They're just making sure that you have enough antibodies that could still protect you in the event of an actual hepatitis exposure. Transmission of infectious agents can be life-threatening to select populations, making screening very important. So for example, pregnant women for rubella and cytomegalovirus and transplant donors for cytomegalovirus CMV negative transplant recipients should receive tissue and blood from a CMV negative donor. Congenital infections. Helpful to diagnose congenital infections that were acquired in utero in a newborn. If the mother is non-immune, they may not see symptoms, but the fetus may experience harmful effects. Examples are Toxoplasma gondii, toxoplasmosis, so that's the transmitted by the cat feces. Treponema pallidum is syphilis. Viruses like rubella, cytomegalovirus, and herpes simplex virus. If the mother is non-immune, so the mother doesn't have enough antibodies, torch agents most commonly found by testing for Toxoplasma, rubella, cytomegalovirus, herpes, Treponema pallidum species, subspecies pallidum. IgM testing on neonatal serum method is of choice for serological diagnosis of congenital infection by one of the torch agents. Antigen detection. So antigen detection can require a significant length of time to become positive. Direct antigen tests are less sensitive than culture, but they have a quicker turnaround time. They're based on antibody and antigen interactions, and indicators on antibodies can be interpreted. So this is the structure of an antibody monomer. So you see, like you've seen previously, you have the antibody binding region, and then antigens bound to those antibody binding regions, or antigen binding regions. Agglutination assays. We will be performing these in this class. So in agglutination assays, agglutinating antibodies react with antigens on the surface of microscopic particles to form visible clumps. Tests can be performed in the test tubes, but are typically performed on the surface of glass, plastic, cardboard slides. Direct agglutination, natural carrier particles, so example, red blood cells, we can do direct agglutination. Indirect agglutination, we have artificial carrier particles, like latex beads that are coated with antigen. Reverse, or coated with antibody. Reverse passive agglutination uses antibodies attached to particle which detects the antibodies, so latex beads coated with antibody. So in the latex agglutination, you're going to have a bead, here's the bead, which is coated with antibody. And when that's coated with antibody, you're going to put the patient's sample in there, and if the patient's sample has the microorganism, you're going to see some clumping. So positive would be clumping, negative would be milky, it wouldn't be clumping at all. This just shows you what the latex beads look like. A positive control would be a solution containing the known antigen of interest. A negative control would be a solution that does not contain the antigen of interest. And a latex suspension control to detect the presence of nonspecific agglutination reactions. So here you can see the positive control, what that looks like, the negative control, and then the specimen or the patient. So you can see it's positive for this one and negative for the others. So if the test latex is positive and the control latex is negative, the control is okay. For the negative antigen control, they should both be negative. That means it's okay. Patient A has a positive for the test latex and negative for the control latex, so that's positive. Patient B has negative and negative, which means it's a negative test. Patient C turned positive for both, so that means that there's nonspecific agglutination happening. So this is a diagram of coagulation reaction with whole bacterial cell antigen. So here's the whole bacterial cell, here's the latex bead, staphylococcal cells with the antibodies, and you can see this interaction with the antibody and the antigens on the bacterial cell. And this is a card showing positive and negative results. So you can see the clumping here, which is positive, and you can see here, those are negative. And this is a comparison of particle agglutination methods that you can review. You can pause the video and take a look at this table. Labeled immunoassays. So immunofluorescent assays allow for the rapid antigen and antibody detection. Monoclonal or polyclonal antibodies are conjugated with fluorescent dyes. And if we have these antibodies, we can wash the antibodies over a fixed patient specimen, and if they stick, we can use a fluorescent microscope to visualize them. So if we see our stuff that's glowing, then we know that it's stuck and it's positive. So the antigen of interest is fixed to a glass slide. Antigen of interest would be from the patient specimen. We can use ethanol, methanol, formalin, or acetone to fix the patient specimen to the slide. And then antibodies that are conjugated to a fluorescent tag are going to bind to the antigen. So we use these manufactured antibodies that are linked to a fluorescent molecule. If the antigen was present in the patient specimen, those antibodies that we created will bind to that specimen and then it will fluoresce. In an indirect fluorescent antibody test, the primary antibody binds to the antigen, and then we have a secondary antibody that's linked to the fluorescent tag, and that binds the antibody. The reason why we have indirect fluorescent antibody testing is the indirect one is a little bit more specific. So if we do get a positive result, it's more specific. Direct would be more sensitive. So here you can see direct. The antibody that's fluorescently labeled is going to bind directly to the antigen. In indirect, we have an antibody binding to the antigen, and then we have these secondary antibodies that are labeled binding to that primary antibody. And this is what DFA stained cells would look like. So if it's fluorescing, that means that it's positive for whatever we were looking for. Sometimes described as enzyme-linked immunosorbent assays, or ELISAs, EIAs are similar to DFAs and IFAs, but they use enzyme conjugated antibodies. So instead of the antibody being stuck to a fluorescent molecule, the antibody is stuck to an enzyme. And when we add substrate, the enzyme chops apart the substrate, producing a color change. Popular and widely used for serological diagnosis of infectious diseases. There's many commercially made kits available, and tests are easily adapted to automation. Objective interpretation of results with colored end products that can be read spectrophotometrically. So solid phase immunosorbent assays bind antibody to a solid phase or matrix, like a microtiter tray, a plastic bead, or a magnetic bead, and then we put the clinical sample in. Then we have a secondary antibody that's conjugated to an enzyme. In the direct sandwich method, the secondary antibody conjugated to the enzyme is added. In the indirect sandwich method, a secondary antibody is non-conjugated, added, and washed, and a third antibody conjugated is added. Enzyme-specific substrate is added, followed by the stop reagent to block enzyme activity, resulting in color end product. So here is what you would be looking at. Here you have a plastic well that's coated with antibody. We know what this antibody is against. So let's say this antibody detects the antigens of HIV. Then what we're going to do is add our patient sample. Patient sample may or may not contain antigens, but if it does contain what we're looking for, then what's going to happen when we add the patient sample is this antibody that was on the well is going to bind to the patient sample. Then you have an enzyme conjugated antibody. So you have this antibody that is conjugated to an enzyme, and we're going to add that in there. Now, if the patient sample bound to that first antibody, this second antibody is then going to bind because the antigen is still there. Now, we add the substrate, and when the substrate is added, that enzyme that was on top of the antibody is going to cleave that substrate and produce a color change. Over here, you have a bead, and very similar to what happened in the well, you have a bead that you expose to the patient specimen. If the patient sample is positive for whatever you're looking for, then it'll bind. You add that second antibody, and the antibody is going to bind to whatever complex is there. And then you add the enzyme substrate. So when you add the substrate, the enzyme is going to cleave it and produce a colored end product. Direct versus indirect. So in a direct, you're going to have a well, or in both of these, you're going to start with a well that has antibody in it. Then you're going to add the patient's antigen, or the patient sample which contains the antigen. Then you're going to add this antibody that is labeled or affixed to an enzyme. In the indirect method, you're just adding another antibody. In the direct method, since you added the enzyme linked antibody right away, you're going to add the substrate, which produces a color change. In this example, you have the primary antibody, secondary antibody, which is linked to the enzyme, and then you add the substrate and it will produce the color change. This one is more sensitive, it'll detect more things, but it has the potential for a lot more false positives. This one is more specific, so this one will, it's harder to get this one to turn positive. So this is just an example of a microtiter plate and strip. So here's a plate where you can have different concentrations of the antibody to determine the concentration necessary, and then here's a strip that we can use as well. Indirect solid phase EIA for antibody detection. So first we put the antigen on the bottom of the well, then we put the patient's serum in there. If the patient's serum is present for the antibodies against the particular antigen we're testing for, then it'll bind. Then you have a secondary antibody that's linked to an enzyme bind, and then you add the substrate to produce the color change. So in this example, it's different because now we're starting with known antigen. This is commercially produced known antigen. When we put the patient's serum on here, we don't know if the patient's serum contains antibodies against that antigen or not. If it does contain the antibodies against the antigen, that means that that patient has been exposed sometime in their lifetime. If it does, it'll bind, and then we add that secondary antibody, and that will bind to the patient's antibody, and that's linked to an enzyme, which we add the substrate, and we can see the color change. IgM antibody capture ELISA is very similar, except IgM is a much larger antibody, so you can see it's a pentamer, it's huge, and you can see it's a very similar process. Membrane-bound immunoassays requires flow-through and large surface area, nitrocellulose, nylon, and others. Procedure highlights it uses a disposable plastic cassette consisting of antibody-bound membrane. Antigen containing clinical sample is placed into the small chamber and evaluated for binding. It allows for the rapid detection of these organisms listed here. And this is just an example of the Strep A kit. And another Strep A. This is a membrane-bound test system. Immunochromatographic test. Clinical specimen is placed in buffer. Process sample is added to the membrane. The buffer is carried laterally along the membrane by capillary action. Labeled rabbit antibody becomes suspended in the buffer. If the antigen is in the clinical specimen, an antigen-labeled antibody latex is formed, and it results in visibility on the test line. The control will capture the labeled rabbit antibody and become positive, and that just ensures that the buffer has migrated past the line and confirms the negative result. The purpose is to detect the antibody in clinical specimens such as anti-HIV. Staphylococcal protein A, labeled with colloid gold in the kit, binds to the FC portion of the IgG with any specificity in the clinical specimen. Resulting complex is carried to the test line, which contains HIV antigens. To detect antibody in a clinical specimen such as anti-HIV, the control line should be positive. It's very popular in physicians' offices, and kits are available that test numerous substances, even over-the-counter kits. So this is something like what you'd see with the COVID test. Basically, you put the specimen in here, and it's going to be wicked up through capillary action. And as it goes up through here, the control line will turn positive because it captures that antibody. But the test line will only turn positive if the antibody that we're testing for is present. The use of serologic testing in specific diseases. So we're not going to go through too much specific detail in this course since this is clinical microbiology and not intro to immunology or basic immunology concepts. So we're going to save a lot of the details for that class. But this is just examples of serological tests for diagnosis of infectious diseases. So you can see the test type and what diseases are detected by that test type. And here's just another arrangement of different types of tests and how they're detected. So if you're curious, you're welcome to take a look at these two tables. You will not be tested on these two tables in this course.